Have you ever wondered how the human brain, the most intricate and complex organ known to us, actually begins to form? This is not a story of years, or even months. It's a journey that starts just a few weeks after the first spark of life. By the third week of embryonic development, a time when most people are completely unaware they're even pregnant, the very foundation of our mind is already being meticulously built. Let's rewind to around day 15 post-fertilization. The embryo, a tiny cluster of cells, organizes itself into three distinct layers: the ectoderm, mesoderm, and endoderm. Think of these as the primary blueprints for every part of the human body. Our focus is on the outermost layer, the ectoderm, because this is the magical source from which both our brain and our entire spinal cord will emerge. It's the cradle of our consciousness. But how does a simple sheet of cells know to become something as sophisticated as a brain? The answer lies in a beautiful cellular conversation. Deep within the embryo, a structure called the notochord sends out precise chemical signals. These signals are proteins, with wonderfully whimsical names like Sonic Hedgehog and W&T, that act as messengers. They travel to the ectoderm and deliver a critical instruction. It's time to become the neural plate. This flat, specialized region is the very first architectural draft of our nervous system. What happens next is a marvel of biological origami. Between day 18 and day 28, the edges of this neural plate begin to rise, folding inward to create a groove. These folds then meet and fuse together, sealing themselves into a hollow cylinder. This is the neural tube, the fundamental structure that will give rise to our entire central nervous system. This process, known as neurulation, is one of the most critical steps in our development. Any disruption here can have profound consequences, which highlights the delicate nature of this early life stage. Once the neural tube is formed, the real construction of the brain begins at its front, or anterior, end. This section rapidly expands and swells, forming three distinct bubble-like structures. These are the primary brain vesicles: the forebrain, the midbrain, and the hindbrain. Each one is a progenitor, an ancestor destined to evolve into the complex regions of the fully formed brain we recognize. They are the initial broad strokes of the masterpiece to come. Then, by the fifth week, this developmental process becomes even more refined. The three primary vesicles undergo a further division, splitting into 5 secondary structures. The forebrain divides to create the components that will become our magnificent cerebrum, the seat of thought, language, and personality, and the thalamus, our brain's central relay station. The midbrain remains, and the hindbrain develops into the regions that will form our cerebellum, responsible for balance and coordination, and the brainstem, which controls our most basic life functions like breathing and heartbeat. Every part of the adult brain can trace its lineage back to these 5 initial segments. Simultaneously, another incredible event is unfolding. A unique group of cells, called neural crest cells, detaches from the edges of the closing neural tube and embarks on a remarkable migration. They journey throughout the developing embryo, differentiating into an astonishing variety of cell types. These versatile travelers will form parts of our facial bones, the nerves in our head, the pigment cells that give color to our skin and eyes, and so much more. They are the pioneers that help build the intricate landscape of our head and peripheral nervous system. By the 8th week, the brain enters a phase of explosive growth known as neurogenesis. Inside the developing brain, precursor cells begin to divide at an astonishing rate, creating billions of neurons. These newly born neurons then migrate to their designated locations and start forming connections, or synapses, with one another. It's during this time that the very first sparks of electrical activity flicker to life within the fetal brain, the earliest whispers of the neural symphony that will one day orchestrate our every thought, feeling, and action. It's crucial to remember that this entire intricate process is incredibly fragile. Every single step, from the first fold of the neural plate to the last synaptic connection, is influenced by a delicate interplay of factors. Proper nutrition, especially nutrients like folic acid, is vital. The genetic code we inherit provides the fundamental instructions, but the environment in the womb plays an equally important role in shaping how that code is expressed. This journey is a testament to the exquisite balance required to build a healthy brain. From a simple, flat sheet of cells to the extraordinary organ of thought, emotion, and perception, the story of our brain's development is truly the story of what it means to be human. It's a journey of breathtaking complexity and profound beauty, happening silently within the first few weeks of our existence. The architecture of our mind is the greatest wonder of the natural world. Thank you for joining us on this incredible journey into the origins of life. If you found this story as fascinating as we do, please subscribe to MedLifeLink. We're dedicated to bringing you trusted, research-based stories of life, science, and medicine. Hit that notification bell so you won't miss our next exploration. See you next time.